Hello, everybody. My name is Dallin Thewison. I'm excited to be here with you today to do this presentation, this video. So let's kind of get started. So in problem number one, we're asked to cross these two organisms, this one right here, as well as this one right there, into different variations or different genotypes. And so what we're going to do is we're actually going to take each of these separate genotypes, and we're going to do a Punnett square. Unfortunately, I already did most of the work, so we're just going to do one as an example, and then we'll kind of get into the rest of it. So for right here, we have heterozygous A and heterozygous A. So when we cross these over, we're going to get a homozygous dominant A. We're going to get two heterozyguses, and we're going to get a homozygous recessive right there. And so when we look at this, so for the homozygous dominant, we're going to have 1/4 because there's only one box. So if we were to highlight this, there's only one right there. If we were to do these ones, well, there's two of them, so let's go 2/4. And if we were to box the last one, we get 1/4. And so that's what I did with all the other ones. But just to save some time, we won't go through all of them. And so the first problem asked is, what is the probability of getting an organism that has this type of genotype? And so what we're going to do is we're going to take the proportions that we've got, what we received right here. So a heterozygous A is 2/4, and then we go homozygous dominant is 2/4. We get 1/4, and then we get 2/4. And I believe we get 2/4. And so when we multiply this all out together, we come out to be 1/128, or we could say 0.78%. So that's our answer to the first one. For B, we're going to do the same process. We're just going to be looking at this model that we've created. And so this is going to be 1/4 from right there. B is 1/4. Oh, sorry. This is supposed to be a 4. Homozygous recessive, C is 1/4. A dominant is 2/4 for D, and the last one is 2/4 as well. And so when we multiply this all out together, we get 1/256, which is equal to 0.4%. And so we'll do the same thing with this one. And so for A, we get 1/4. I really need to work on my fractions. B, we get 2/4. C, or homozygous recessive C, is 1/4. Homozygous recessive D, well, if we look right here, it's 0. And so it's 0/4. And if we were to do the last one, we don't really need to anymore because we know what this is going to turn out to be. But this is 2/4. Now, since there's a 0 and when we multiply, there is a 0% probability that we'll ever have this, or you could say it's impossible for us to have an organism with this type of genotype. Now for the last one, we just do the same thing. So we get 1/2, or 2/4. That's equal to 1/2. 2/4 as well. This is going to be 1/4. Let's see, did I do this wrong? I did. This is actually supposed to be that. So we get 2/4 in this one and 2/4 in this one. And I believe those are all correct. Actually, this is supposed to be homozygous. So sorry, I wrote the problem wrong. And then what this equals is 1/32, or 3.1% chance that we'll have this type of genotype. So that answers question number one. Hopefully that was informative. Now let's go into question number two. So most of these questions we were able to find from the reading that we did in the book, or in the PowerPoints, as well as the instruction notes that we received. And so A asks, a paired homologous chromosome are sometimes called, well, the answer is bivalent or tetrads. And we'll kind of get into those a little bit more. I'll show an example because one of the questions asks a little bit more about that. Question B is the structure that connects a centromere to the microtubules of the meiotic spindle is called a kinetochore. And once again, that definition is just in the reading. And then C, I can actually do a little demonstration for C. So we can kind of see a visual of what it is. Let me solve this really quick. Put this back. Okay, so see, we have a diploid fruit fly that has 4 pairs of chromosomes. So if we were to draw this out, we have 1, 2, 3, and 4. A fruit fly cell in prophase 1 has how many sister chromatids? Well, in prophase 1, we know that these chromosomes have doubled, in a sense, have copied itself into the exact sum. So this one is equal to that one, and so on and so forth. So the question is asking, how many sister chromatids do we have in this? Well, we can conclude that for each of these chromosomes, so one of these is equal to 2. So since we have 4 right here, so 2, 4, 6, 8, 10, 12, 14, 16, the answer to C part A is 16, 16 sister chromatids. Then while a fruit fly cell in metaphase 2 has how many sister chromatids? Well, metaphase 2, we know that after meiosis 1, these chromosomes right here go down over here into one cell, and these will go into another cell. Well, what happens is these 4 chromosomes, if we were to draw them, probably can't see that. Anyways, there's 4 of those right there. We're asking how many sister chromatids we have. Well, since there's 4 of these, so we can go 4 chromosomes times 2 sister chromatids per chromosome, we end up with an answer of 8. The next question, part D, is test crossed. That's the answer. Is a type of genetic cross that can determine if a purple flower pea has a homozygous P dominant or a heterozygous P genotype. And this is what Mendel kind of discovered as we went through his readings. And so just kind of more like a definition of what he discovered. The next answer is in E, I should say, for E. A non-functional allele is sometimes called a null allele. And I'll go through these in a second. While a partially functional allele is called a hypomorphic allele. And so these are different because in a null allele, the gene is not being expressed at all. While in a hypomorphic allele, there's a little bit of expression, or just a partly functional allele that is making proteins and whatsoever. So that's the answer to that one. And the last one for part F is Mendel's law of independent assortment. Well, independent assortment involves 2 traits, while Mendel's law of segregation involves a single trait. And we find this just in the reading that we do. So hopefully that helps a little bit. So let me erase this really quick, and we'll get into number 3. Okay. So number 3 asks, so we have an organism with a TT heterozygous R and a heterozygous Y chromosome. And each of these alleles or genes alleles, I should say, assort independently. Now the word independently is very key as we are going to solve this first problem. Now, let's see here. So in part A, it says diagram all the possible homologous chromosomes arrangements in this organism during metaphase 1. Now metaphase 1, we have to remember that it doubles itself. Or it's already gone through prophase 1, so it's already made a DNA replication of itself, and it's kind of going towards that metaphase plate, where it's lining up on that. And it says, be sure to indicate which of those are found on each pair of homologous chromosomes. And so what we have to realize is that independent assortment, you can only have homologous chromosomes. I'm forgetting the word right now. Anyways, so we have T, so this is going to be the first one. and T. So this is them arranging along the metaphase plate. And then we have our homozygous chromosome, R, and then little r, and then we can also have a homozygous dominant and homozygous recessive. And so this is one possibility. The next possibility, we do the same thing. T, and then we can actually just swap the R's. And so we're gonna get the homozygous recessive right there, homozygous dominant right there, and then we can just keep the homozygous dominant Y and the homozygous recessive Y. This is a Y. These are little Y's. And then we can also have, so if we were to do another one, so we line it on the metaphase plate once again. This time we're just going to keep the R's in this pattern. And so what we'll do is we'll have R, and little r, and homozygous recessive R. And then we'll have a little y, or homozygous recessive Y, and a homozygous dominant Y. Well, there's one more possibility that we can have, actually, and it's taking these R's and then swapping those Y's. So if we were to draw this again, we get two T's, or homozygous dominant T's. We get, let's see here, homozygous recessive R, homozygous dominant R. One is a homozygous recessive Y, and a homozygous dominant Y. So as you see right here, there's 4 different possibilities of these arranging along the metaphase plate during independent assortment. So that answers part A. The next, it says, indicate all the possible gamete genotypes that can be produced in the organism at the conclusion of meiosis II. Now, I'm going to erase this really quick so I can draw it again. So we're going to keep those right there, those genotypes. So we have to remember that in meiosis II, these alleles have already, so remember that in prophase 1, they doubled. And then from these, so this is just one genotype, right? But you have to imagine that it's being applied for all of them. So these will break out into 2 cells. So after meiosis 1, these are 2 cells. And then during meiosis 2, both of these T's are going to be become gamete or haploid cells. So what we're going to do is we're going to take the possibilities. Now, since the homozygous or dominant T's are the same, we only have to really worry about one of those. However, with the R's and the Y's, they're heterozygous, and so there's multiple combinations that we can have from them. So for example, we can have a T and then an R, a big R, and a big Y. We're going to have a T there. We can have a little r and a big Y, and we can have, once again, a big R and a little y, and the last one is little r and little y. And so there's 4 possibilities of gametes that can be produced from these genotypes. The next question is, how many sister chromatids are found per cell in the organism during metaphase 1? So metaphase 1 is when we have, so metaphase 1 is when after prophase 1 they've all doubled. So right now we have T, let's say R, just an example, big Y, and then we have homozygous T, homozygous recessive R, and homozygous recessive Y. Now, for each of these, there's 1 sister chromatid per each of these. So this one has 2, this one's gonna have 2, this one's gonna have 2, this is gonna have 2, this is gonna have 2, and this is gonna have 2. And so if we add all those up together, we get 12 sister chromatids. And then during metaphase 2, how many do we have? Well, metaphase 2, or it says how many chromosomes are found per cell in an organism during metaphase 2. Well, as in this demonstration, we just took the T's, but we have to remember that there are the R's as well as the Y's in these. And then so when they separate out, we'll have, so in these combinations, there's only going to be one chromosome, there's another one, and then there's another one. And so the answer is going to be 3 chromosomes. Hopefully that makes any sense. But anyways, so we're going to question number 4 now. I don't know how long I've gone. I'll try and hurry. Okay, so question number 4 says a horse has 32 pairs of chromosomes. How many chromosomes are present per horse cell at the conclusion of meiosis 1? So we have to remember that there's 32 chromosomes. So part A is asking how many are there at the conclusion of meiosis 1. So meiosis 1, we have to remember that during prophase 1, it jumped from 32 and it copied itself. So we're going to times that by 2, right? So this is going to give us 64. Well, at the end of meiosis 1 is when it breaks apart and it kind of divides itself in half. And so we're going to divide this by 2, and so we're only going to get 32 chromosomes. Then in part B, it says how many tetrads are present per horse cell during prophase 1 of meiosis. Now this goes back to one of the definitions that we were talking about. With the definition of a tetrad, it talks about a paired homologous chromosome. And so this is a chromosome, right? Well, this right here is a tetrad. And so one chromosome, this is one tetrad. Or you could say a bivalent. It's the same thing. And so let's see here. What was the question again? So how many tetrads are present during the process, prophase 1 of meiosis? Well, in prophase 1, there's 32, right? Well, it doubles itself and it equals 64. Well, we have to remember that for every chromosome, in order to be a tetrad, it has to have a double in a sense. And so the answer actually is just going to be 32 tetrads because a tetrad is just like it's a paired homologous chromosome. And so we don't really need to times it by 2. It's just when we go through prophase 1, it times it by 2, so we actually get our tetrads from that. Let's see, how many sister chromatids are present per horse cell during meiosis or metaphase 2 of meiosis? Now, we have to realize that once it's already gone through, so it's already split itself in half. And so it's gone from 64 chromosomes, right? So I'm just going to write this. So we've gone from 64 chromosomes and we've divided by 2. So we're now at 32. So we have 32 chromosomes currently at metaphase 2, but we're asking for sister chromatids. Now, with the definition of a sister chromatid per chromosome in a diploid cell, we see that, let's see here, we just have to times this by 2, so times this by 2, and that gives us 64 sister chromatids. Let's see, the last one is how many bivalents are present per horse cell during anaphase 2 of meiosis. Now, once again, bivalent is when we have 2 chromosomes that are like each other, or homozygous, 2 homologous chromosomes, sorry. And since at the end of anaphase 2, we're only left with, so if this was to divide, we come out to be, we have 1 cell with that, 1 cell with this, another cell with this, another cell with this. And so if we look at this, there are no chromosomes in it, they're just sister chromatids. And so based off of the definition, there are 0 bivalents after anaphase 2. Okay, so that answers number 4, and we'll get into question number 5 really quick. Okay, so we have this pea plant, it's TT, or heterozygous T, heterozygous R, homozygous recessive Y, and a heterozygous A. And it's being crossed with heterozygous T, heterozygous R, and heterozygous Y, and a homozygous recessive A. Now, with each of these genotypes, they have phenotypes that are associated with them. And so we know that a big T, TT, or a T little t, these are going to be tall plants, just based off of the information that's given us. With a little t, this is going to be a dwarf. And now if we go to R, and heterozygous R, these are going to be round. And then the little homozygous recessive R, well, that's going to be wrinkled. And so we can kind of see the pattern with that. I should probably finish these off really quick. So, but actually, I'll get into that a little bit different. So you can kind of see that because the heterozygous, the big T is dominant. There's a glare right there. Try to fix that. I don't know if you can see it. Anyways, since the heterozygous is dominant, it still has that tall genotype that are in it. The pea plant is still going to be tall. And so it's going to help with the probability as we kind of get into this. And so question letter A asks, what proportion of the offspring will be tall, round, green, and terminal? Well, we need to get into our proportions or find out how many homozygous dominants there are going to be, how many heterozygous T's are going to be, and how many homozygous recessive T's there are going to be. And so let's turn into those calculations. And so we'll do T's. So T, and then little t, and then we have R. You just do this really quick. The next one we have Y's. There's a heterozygous Y. And then the last one that we're doing is A's. So homozygous A, dominant, and then heterozygous, and then recessive right there. And so let's turn into these proportions. Sorry, I need to have my equal signs. So if we were to take this is a heterozygous by heterozygous, and so we're going to get 1/4. This is going to be 2/4. And this is going to be 1/4. Well, we got the same thing, heterozygous, and so 1/4, 2/4, and 1/4. Here we have a homozygous by a heterozygous, and so there's not going to be any right here. However, we're going to have 2/4 and 2/4. And with the last one, we have heterozygous by a homozygous recessive. So there's not going to be any homozygous dominant. However, there's going to be 2/4 for heterozygous and 2/4 for homozygous recessive. And so the question is asking, what is the proportion of the offspring that will be tall, round, green, and terminal? Now we need to realize that to be tall, you have to add these together. And so we get 3/4. And then I'll just do these really quick. We get another 3/4 for the round. Because we're including those. We have green, 2/4, which is a recessive phenotype. And then heterozygous A, which is 2/4. And so if we were to multiply these out together, we get an answer to be 9/64, which is equal to 14.1%. Now with part B, we do the same thing. And so just to hurry and do it really quick, we're asking for proportions of a, like, let's show this down real quick. So we need to find the probability of the genotypes of the homologous recessive T, heterozygous R, heterozygous Y, and heterozygous A. Make sure I did that right. And so if we're going to look at here, so we have 1/4, we have 2/4, we're multiplying these together, heterozygous Y, so we get 2/4, and the same thing with heterozygous A, so we get 2/4. And so if we were to multiply those out together, we get 1/32, which is equal to 3.1%. So for part C, if we do that one now, so we want to find the proportion of the offspring that will be heterozygous for all genes. So heterozygous is going to be, like, for example, this one, and whatnot. So let's do that really quick. So heterozygous for T is 2/4. We multiply that by 2/4 for heterozygous R. Heterozygous Y is 2/4 as well. And then heterozygous A is 2/4. So we multiply those out together, we end up to be 1/16, which is equal to 6.3%. And the last one that we're going to be doing is what proportion of the offspring will be dwarf, wrinkled, yellow, axial. And so we just kind of do the same things. And so to be dwarf, we need to be recessive, and so that's 1/4. Now, to be wrinkled, we need to have a recessive, or homozygous recessive R, and so that's 1/4 as well. And then the last ones are yellow, which is homozygous. Actually, yellow is going to be the heterozygous, so we get 2/4. And then the last one is we need to be axial, and that will be, as it says in the instructions, is dominant. And since we have 0 right there, but we have 2 right there, we just multiply by 2/4, and this will equal out to be 1/64, which is equal to 1.6%. Anyways, so that answers all the questions. Hopefully you were able to kind of see my writing, as well as my just thought process. But anyways, until next time.